welcome to this National MS Education and Awareness Month conference hosted by MS Focus, the Multiple Sclerosis Foundation, and made possible with support from Biogen, Genentech, Novartis, EMD Serono, Sanofi, Viatris, and Bristol Myers Squibb. I'm your host, Deborah Foreman, the Educational Programs Coordinator for MS Focus, and I'm joined by Dr. Deborah Backus, who will be discussing the importance of clinical trials. After she's finished, we'll open it up to your questions and comments. Now I'm delighted to introduce our speaker. Dr. Backus is a grant-funded researcher, physical therapist, and educator with more than 35 years of experience in the neurorehabilitation field. In addition to serving as Vice President of Research and Innovation, she is the Director of the Virginia C. Crawford Research Institute and Director of the Multiple Sclerosis Research at the Shepherd Center in Atlanta, Georgia. Her research has been funded by the National Institutes of Health, the National Institute on Disability, Independent Living and Rehabilitation Research, and the National MS Society. She is currently principal investigator for one of the largest exercise trials in MS, comparing the effectiveness of an exercise program delivered in the facility or in the home. Dr. Backus's mission is to empower patients, students, and colleagues to achieve their greatest potential and to advance rehabilitation science and care for people with disability due to neurological injury or disease. Dr. Backus, thank you so much for being with us today, and I'm going to turn it over to you.
Well, thank you so much. Thank you for that introduction, and thanks all for being here today. I'm going to go ahead and share my slides. And there we go. So I'm really excited to talk with you today about clinical trials. I believe that at the key of all our clinical trials, all of the research that we do are the people that we serve clinically. And that we can only be successful with our clinical trials if we engage the people we're trying to reach. And in this case, if we engage the people with multiple sclerosis. So I believe clinical trials are an opportunity for us all to change the impact of MS. So show of hands, how many of y'all have used aspirin or painkillers? I suspect there's a lot of you that are raising your hand. How many of you have gotten an MRI or other imaging of your brain? Probably many of you have had that as well. Many of you have partaken in medical treatments, drugs, have used devices, have undergone diagnostic procedures, all of which are possible, all of which are safe and effective in accomplishing their respective tasks because of research. Now, research has been going on for a very long time. I thought this was really interesting. This is one of the earliest accounts of research at 500 BC. It's reported on not in a scientific journal, but actually in the book of Daniel in the Bible. King, and I tried to figure out how to say this, Nebuchadnezzar, King N, was a military leader, and he wanted the young men who were working for him to be very strong. So he decided he was going to put down an edict that they should all have meat and wine in their diets. And there was a group of them, amongst who was Daniel, who said, no, we're vegetarians. Now, whether or not they used the term vegetarian, I'm not entirely certain, but they said they didn't want to eat meat and wine. They wanted to have vegetables and water. So the king let them do that. And shortly after this, he noted that those that had been eating the vegetarian diet and the water seemed healthier than those that were having the meat and the wine. And so he published this and actually changed what at that time, I don't know if they called it health policy either, but basically changed health policy and said it was okay for people to have a vegetarian, or his soldiers to have a vegetarian diet and drink water. Now, King N, that is one way of thinking about research. And he did, by all accounts, observe two different behaviors. but he really didn't take the most scientific method. So when we consider what research really is, it needs to be a very careful, rigorous approach to examining a question. And there's a whole procedure that's put around that. And the purpose is to make sure that we really understand that what the decision we're making from that exploring is actually valid, and that we can feel confident we can apply it to the people that we want to serve. So, you know, King N, he did compare one diet to another, but he had wine and meat compared to vegetarian diet and water. If he really wanted to know which variable was having an impact, he might have done meat and water compared to vegetarian and water. He also might have very rigorously decided who was getting one versus the other. He really just kind of had what's called a sample of convenience. And he didn't put any parameters about time of day that people were eating, how many meals that they ate, what kind of exercise they had. So we really can't know how those variables affected. So research, we want to try to collect information on a particular question in a very rigorous fashion. And there's what's called the scientific method to doing that. And this figure, to me, I'll use it again as I go forward in the presentation. We observe a behavior and we ask a question. And from that question, we're going to make best guesses, which are called hypotheses. So we might say for King N that those people who had a vegetarian diet, they might have been able to have better body fat. They might have been able to think more clearly than those people who had the meat and the wine. And then we can run an experiment. We can recruit people or participants, and we can assign them to different groups, and we can do some tests to see if our guess, our hypothesis, is in fact true. And if we've been very careful in applying the scientific method, we will be able to develop a conclusion or establish a conclusion, what our results are. And we can be more secure in what that conclusion is, the more we've controlled the situation and the more people in which we've looked at that situation. So that's research. Using a scientific method, we observe, we generate questions. Is this better than that? Then we establish a guess about it or a hypothesis. Then we actually test it. And then based on our results, we come up with a conclusion. And only by using this method will we know if something is safe and effective. Excuse me. So when we consider MS research, there are actually a few different ways that we can use this scientific method. One is in basic science. And a lot of research, especially related to drugs or exercise, often will start with animal studies, and that's called basic science. We also have epidemiological studies where we can look at, for instance, distribution of disease. How many people have relapse remitting MS in the United States versus those with progressive MS? What is the distribution within progressive MS of walking ability versus lack of walking ability? We can look at the factors that affect their health. What's the impact of smoking on relapses, for instance? We can also look at how people make decisions about the treatments they receive. Is there a difference in different parts of the countries for people with MS and the kinds of services that they access? Those would be examples of epidemiological studies. There's also patient registries. Some of you may have heard of NARCOMS or iConquer-MS. These are registries where you can input your information about yourself, about your disease and your disease history, and different activities or services that you use relative to your MS and your healthcare. That information then establishes a very large data set where researchers can go in and look for patterns of activity that explain certain outcomes for people with MS. And finally, there's what we're going to talk about a little bit more today, and that is clinical trials. A clinical trial is a trial in human volunteers or taking data or samples from humans to better understand what's happening, to better understand their health or the impact of the disease. They are the way that we find better ways to prevent MS or other health conditions, that we can detect it using different diagnostic tools. So it might be that we're using MRI to detect lesions or CT scans. Might take blood samples to look at other conditions, that we can diagnose it and finding ways to treat it. So with MS, one of the most common things that people think about with treating it is the use of disease-modifying therapies. The way to find out if a disease-modifying therapy is safe and effective is by doing trials in human volunteers. And clinical trials, if we use the scientific method of observing and defining a very careful question, does this drug slow the progression of MS safely? Or does this drug slow the progression of MS more than this drug? Is if we, that is the kind of question we can develop. And then we can develop hypotheses based on what we already know about that drug. And we can make a best guess that drug A might be better for reducing relapses than drug B. Then we can do an experiment to test that, and based on the results, come up with a conclusion. Only by doing this can we find out what does and doesn't work in humans. Continuing to do it in animal studies isn't going to help us because the animals have different nervous systems. They respond differently to different environmental factors, to different drugs. So the only way we can do this, only way we can take the guesswork out of medical care is to do trials that follow this scientific method. A clinical trial, although it can look very much like clinical practice and if done correctly should feel as safe as clinical practice, isn't the same. The questions are a little bit different. When you go to a clinic to get your care for your MS, they're worried that you're getting good quality care that will either help cure or ameliorate your symptoms or improve your situation. They're also interested in the fact that you're satisfied with the care that you receive. In a clinical trial, while the interventions are similar, they may be drugs, we're more concerned about the safety and the quality of the data, that you are participating in a study that protects your rights, no matter what. At the end of the day, the most important thing is that the participant, the patient, their rights are protected, and that, and I'll talk more about the sponsor and what that means, but that the different parties that are involved are satisfied with the outcomes, that they feel comfortable that the conclusions they can draw are valid, that they can believe those conclusions. A clinical trial involves one or more human subjects that will be assigned to one or more interventions. An intervention can be thought of it's a way of intervening, but it's a treatment, which that treatment could be a drug. That treatment could be the use of a technology or a device, or it could be something like physical therapy or an occupational therapy intervention. So it could be exercise, or it could be some training types of interventions. It could be cognitive exercises as well as physical exercises. Oftentimes in a clinical trial, this may include a placebo or a control. A placebo is when it's a sham or there's no therapeutic value. And you may have heard of this referred to as when people are doing drug trials, they used to talk about the sugar pill versus the real pill. And they look the same, but one actually can impact your body and the other one can't. So the sugar pill would be considered a placebo. It doesn't have any therapeutic value, but it looks like the actual treatment pill that you might be given. Sometimes that's done with electrical stimulation therapies. So using surface electrodes, placing a pad on the brain, on the head to activate the brain, or on the muscles in the arms or the legs, you can apply stimulation. In some cases, you can have a placebo where the electrodes are there, but there's no stimulation provided, or a low stimulation so that it doesn't have the same intensity as the one that has what's called therapeutic value, that's going to impact your body or your nervous system or your muscles. A control group is a group to which something is compared. Sometimes a control group could be a placebo. So giving the placebo pill and comparing it to a treatment pill. Sometimes the control group is one drug and they're comparing it to another. So if there's a drug that's been around for a really long time, a new drug might be compared to it when it's considered a gold standard. So a great example of that is aspirin. For a long time, aspirin was the painkiller. But then other drugs came along and they were compared to aspirin. So aspirin was the control group, but the new drugs for pain would be the treatment drug. Clinical trials are done when there is assignment to a control group and a treatment group. That assignment is done randomly. That means that instead of letting a person choose, like King N did, or telling them which group they would be in just based on their characteristics, you're using some other way of randomly assigning them. So sometimes that might be choosing from a stack of envelopes, and in the envelope it'll say which group you're in, either the control group or what's called the treatment group. Or there's what's called random number generators. So using a computer, and some of y'all may know about this, they'll generate a list of numbers and it'll say 1 is treatment group, 2, 3, and 4 are control group, 5, 6 are treatment groups, 7, 8, 9, 10 are control groups. And then as the participants come into the trial, that order of numbers determines which group they're assigned to. The reason that's done is you don't want anything to influence or bias the findings. So with King N, people picked what they liked. And so they were going to get the kind of that influence of them doing what they like or what they prefer that can influence how they respond. So if you make somebody who doesn't like to eat meat, then they may be very unhappy, which can actually influence their health. So that would bias those findings. If I look at somebody and I say, oh, this person is obese, they really need to go into this diet where there's fewer calories and less fat, that person is likely to benefit from that because I've picked them because they have the opportunity to lose weight or to be healthier. So I've biased that selection. So a randomized controlled trial is a clinical trial in which people are randomly assigned to either an intervention or a control situation. And that control situation might be another intervention, or it might be no treatment at all. Another aspect of a clinical trial is that either the personnel or the people in the trial who are running the trial or doing the assessments are blinded, or the participants are blinded. So I'll first start with the sugar pill. The idea with the placebo, having a sugar pill look just like a pill that you're testing the outcomes of, is that neither the participant, the person who's taking it, nor the researcher knows which one they got. And so there can't be any bias. So you don't want the person being influenced by thinking they got the treatment one versus the non-treatment one and having that impact how they feel about getting that treatment. That's called they're blind to what they're receiving. In a blind trial, that's typically when you're talking about the researcher not knowing which intervention the participant, the patient got. That way, when I look at the data, I'm not biased and I don't say, well, I really believed that treatment A is better than treatment B. And obviously, all those people that got treatment A are better. We don't want that influence of the researcher's bias towards one or the other of the interventions. A double-blind trial is when neither the patient nor the researcher knows which intervention they're giving. No one knows what drug they got. Did they get the placebo or the controlled drug, or did they get the actual treatment drug? And that way, no one's biased. You just kind of know this is the response I've had to this drug, and this is the response I'm seeing that patient has to this drug, and that's what the outcome is. And there's no influence of what I think should be happening or what the patient thinks should be happening. And then there are specific enrollment criteria. And what that means, enrollment criteria are the rules for accepting somebody into a trial. And the reason that that enrollment criteria needs to be so specific is so that you know that whatever intervention, what therapy, technology that you're using, whatever outcome you get is because of that and not because of some aspect of that individual. So a good example is, for a long time, people wouldn't enroll somebody over 65. Now, don't get mad at me. This is just historically, and I'm going to tell you why. I don't necessarily use this rule, but in trials, when people are looking at the impact of exercise on one's health and walking ability, people used to say they only wanted to recruit 18 to 65-year-olds. And one of the reasons was because the belief that people that were 65 or older are starting to age. And age in and of itself can slow or lessen the impact of exercise. So if you have somebody over 65 who gets a certain exercise intervention and they don't respond, you don't know if it's because that intervention is not effective or if it's because that person is over 65 and they've been aging. When we do some of our exercise trials in people with MS, we may choose to focus on people who are able to walk versus people who use a wheelchair. And the reason is because they have different abilities and they may respond differently. The same might be true for relapsing versus progressive MS. The nervous system is responding differently. So if we give a drug to somebody who has relapsing remitting MS, their nervous system might respond differently just because their MS is different than somebody with progressive MS. So if we really want to know what the impact is of that drug on their MS, we're going to try to make it very specific to the type of MS that they have. It's not intentional. It's not, it is intentional. It's intentional in the sense that it's meant for rigor and being able to make the most sense of the data, not to leave anybody else out. But it's also kind of one of the reasons why it's so hard to enroll people into research studies is because it's very specific criteria, and also why it often takes a really long time to do research that affects the entire population, because we have to have very specific enrollment criteria. And finally, the clinical trial has a very specific protocol with all the study methods. So that starts with the people we're enrolling. It starts with, it then goes to the timing of all of the activities in the trial. So if people are supposed to get a regimen of treatment, whether it's a drug or an exercise intervention, a therapeutic intervention, they're supposed to get it for 8 weeks. They should only get it for 8 weeks. If it's only 12 sessions, they should only get it for 12 sessions. If they only get it for 8, then you're really not testing the 12-week intervention. If you get it for 14, you're getting too much of the intervention. So it has to be very specifically defined so that you'll know that the outcomes that you see, the results that you get, are related to what you did. And it's the only way that you can really answer the question. It also, the protocol will also specifically define the measures. So based on whatever the question is, if the question is, does drug, excuse me, does drug A help people with MS walk faster than drug B? Then the walking outcomes have to be related to walking. That's the only way you can answer that question. Does drug A help somebody think more clearly than drug B? Then you have to have cognitive assessments. And then it's imperative that those get collected as at the right times in the protocol so that you can carefully assess. And that's something that's really important to understand if you participate in a research trial, is that that protocol, the reason we do that is so we don't waste anyone's time. If we don't follow the protocol, then we won't know if what we see is because of the protocol or because we deviated from it. So it's very important for clinical trials to have a specific protocol. I'd like to, let me finish this, and then I'd like to go ahead and see if there are any questions at this point, but let me just finish this, make sure I didn't miss anything. So the protocol defines all aspects of the study, who's going to be in it, who's not, the timing of everything, the timing of when we talk to you and get your agreement to be in the trial, the timing of when you get your assessments, and when you get the intervention, how long, the dosing, all of that is defined. And it is critical because at the end of the day, with clinical trials, we are looking at the safety and the effectiveness of whatever that intervention might be. And again, that might be a pharmacologic or drug intervention. It might be a technology, or it might be a rehab approach, such as physical therapy, occupational therapy, speech therapy. So I'd like to stop there and see if there are any questions before I go any further.
Okay, thank you. So now that we are ready for questions, I'd like to explain how to do that for those that aren't familiar with it. If you have a question or a comment, you can ask it by using the Q&A button in the app, which also allows you to send your question anonymously if you choose, or you can ask your question live by raising your hand. To do so, click the raise hand button or press the number 9 if you're on a phone. I'll call on you, and then you'll have to unmute me. I'll unmute you, then you'll have to unmute to ask your question.
And
so let's see, let's start. Let me take a look and see if we have any to start with. I do have a question. Well, we do have one before mine. What is the benefit of participating in a clinical trial?
So I am going to talk about that. So if it's okay, can I save that question to the end? I am going to cover that a little bit.
Okay.
There's lots of potential benefits, and I'll share with you a couple of outcomes from clinical trials related to MS.
Okay. How about, what do they do with all the data that's, at the end of a clinical trial? What do they do with all of that?
So all of this data that I'm talking about, we're collecting, will be housed. It's de-identified, which means we take off any personal information. We do our analyses, and then we house that data for 7 to 10 years to keep it for future analyses. There's a lot of work in different research funders, some federal agencies, to try to get people to share their data so other researchers can use it. But always the information about the individual is taken out so that you can't be identified from that data.
And if I wanted to, how could I find out about the results?
So more and more investigators are sharing data, reaching back out to patients to tell them the outcomes, but they can't do it until the end of the trial. So they can't tell you your personal findings of, what your response was to a drug or an intervention or how you compare to others. But at the end of the trial, they can share the results with you. They're often published or presented. And so you can also find it that way. But so it could be shared either way, but you are allowed to ask for that data.
And what happens if I'm put into the control or the placebo group? What's the difference between those two groups?
So, so there is the potential to be in the control or the placebo group no matter what, right? So there's a 50-50 chance in any clinical trial if there's a control group. And after this, I'm going to show you some more about the different phases. I really wanted to make sure that you're all okay with the definitions so far. But if you are put into the control group, well, one, you won't know if you're put into a treatment versus a non-treatment group. You just can't know that unless you are in a rehab trial, such as an exercise trial, but you won't know that. And so it is a risk that you're taking, and it is something that you should carefully consider. And I'm going to tell you about the process that an investigator should use to make sure that you're fully informed about what the risk is if you are put into the group that doesn't get the treatment, so that you can consider that and you can discuss that with your providers to make sure that you're not putting yourself in a position that you're not comfortable with. But it is a part of the research process because it's research. And the thing that I like to remind people is that we don't actually know if the treatment group is going to benefit more than you in the control group. So when we're testing a treatment, especially in the earliest stages that I'd like to move on to and tell you about next, in the earliest stages, we don't know if that treatment is better or is safe. So you actually aren't necessarily in a worse position if you're in the control group. And that's why we're doing the research, is to find out about that. But that will make more sense. Let me go ahead and, if there's no other questions related to the definitions of clinical trials, let me go ahead and tell you more about the phasing and some of the other things, and then I will hold on to that question and revisit it again. All right. Thank you for those questions. All right, so clinical trials. Clinical trials, human trials, where we're testing a specific question. We've made a guess about it being safe or efficacious. And by efficacious, we mean, is it effective? Is it doing what we want it to do? And we have to have a very rigorous approach. So we have to carefully define our methods, which including the right people, enrolling them in the trial, doing the tests at the right time, and delivering the intervention as we think it would be the most effective. So in MS, what does that mean in clinical trials? How do we define efficacy? In MS, when we're looking at different drugs, the different disease-modifying therapies, those trials, they're trying to see, oftentimes you'll hear the term decrease or the annualized relapse rate, how many relapses there are a year. If we're doing a trial and we're looking at a disease-modifying therapy, then we want to decrease the number of relapses people have in a year. Another thing that we like to look at is, are we going to slow the progression of disability? Or are we going to decrease the amount of disability that someone has? And in some cases, it'd be wonderful if we reverse the disability. So in our clinical trial, we're looking at exercise delivered in the home versus in a facility. And what we want to see is that people get better with exercise. So we want to help their walking improve, which would decrease their disability. One of the tools that's used to measure that is the Expanded Disability Status Scale, or the EDSS. And some people on this call may have had that done before in the clinic, but it's oftentimes done in research trials as a measure of disability. So it's a scale of 0 to 10, 0 being no detectable disability, even though there's MS, and 10 actually being deaf. But below that, between 0 and 10, is a progression. From 0 to 4, the amount of disability is based on all of the body systems, not just walking, but also cognitive ability, bowel and bladder function. When you get above 4, the distance that you can walk and how much help you need in walking starts to impact that level of disability. So when you get to 5 to 6, you have a slower and you're not able to walk as far because of your MS. When you get above 6, you start to require things like canes or crutches or bracing. And then above at 7, you require a wheelchair for community. So it's kind of this progression along the scale. So what we look for when testing an intervention, whether it's a drug or, as I said, exercise or some rehab intervention, we're looking at, can we slow or decrease the amount of or progression of disability? In many drug trials, they're looking to slow the atrophy or the shrinking of the brain tissue itself. They might be looking for no further evidence of disease activity, or NEDA. And we're asking the question that, compared to the control, compared to the placebo or another drug or intervention, is there fewer relapses, a slowing of the progression of disability, less atrophy, better MR and better activity in the brain? That's what we're looking at when we're looking at efficacy in MS. Now, clinical trials are thought of in stages. They don't always, even though this figure shows it progressing, it doesn't always progress. And actually, there are many trials that start on the left at preclinical, never make it to phase 3, or there's also a phase 4. But clinical trials for the Federal Drug Administration in the United States have to go through this phase, and it's a good thing. Has to go through these phases, because each phase serves a very specific purpose, keeping us safe and making sure that the drugs that we take and the devices that we use are safe and going to be effective for the purpose we think they're being used for. So a preclinical trial is a trial in which it's done in the lab. It tends to be those basic science trials where they're starting to look at the risk versus the benefit. If we give this drug, are there any risks that we need to know about? Typically done in animal studies. Is the drug poisonous? Are there what's called adverse effects or bad effects? Of course, the worst would be if you give a drug to a rat and it dies. Well, that's an adverse effect. Not going to be interested in moving that forward to another phase. Also in those early lab studies, preclinical studies, trying to determine what's the right amount of the drug. In stem cell studies, what's the right injection of stem cells that is safe for the animal? And once we know that, then we can start to calculate and consider what that might be for humans. If something is safe and potentially effective in those preclinical trials, it then progresses to a phase 1 trial. A phase 1 trial is done in humans. It starts in a small group, typically less than 80 to 100 people, often only 1 or 2 facilities. Those facilities might be a clinic or an academic center. And that is specifically to see if doing a similar approach in humans that was done in the preclinical trials in animals is safe, and starting to look at the dosage and see if there's any side effects. If we get past that phase, and it is indeed safe, and we know a range of dosing that could be effective, and there's no significant risks or side effects, can move to phase 2. A phase 2 enrolls more people, up to 300 participants, and we're really starting to now look at things like walking function, disability. We're trying to see, is it effective? Is it slowing down the rate of progression? Is it decreasing the number of relapses? Is it helping people walk better and faster? Then it might be at more centers, and typically it has to be to enroll that many people. We want to enroll more people because the more people that we have, and the more we see the same results, the more confident we can be that that's the outcome we can expect with that intervention. Phase 3 is in a very large group, many centers, to demonstrate the effectiveness. It's when we might start comparing two drugs. So comparing, ibuprofen to aspirin, for instance, to see if there's a better effect of that intervention. Phase 2 is when we are starting to really think about effectiveness. Phase 2 is where you can feel more confident that the intervention, if you're enrolling in a trial, has been shown to be safe. So in terms of the question related to being put in the control group versus the intervention group, at phase 1, you're making a decision that, in fact, the intervention, they're still looking at safety. And so I guess it would really be a 50-50 chance that the control group is the good group versus the treatment group is the good group, quote unquote, good group. So there's no way really necessarily to know that, but they wouldn't have gotten to that point if they felt confident that they weren't going to do irreparable harm. So that's something for you to consider. And in phase 2, they've gotten past those early safety trials and they feel a little bit more confident that there's more benefit than there is risk. Now they're trying to see just how effective it is. How much can it be more beneficial than something else? So those are your clinical trial stages that are pretty traditional, phase 1, 2, or 3. It can take many years to get through all of those phases. It costs a lot of money to get through all of those phases. Phase 4 is when something gets through phase 3, it's been shown to be effective, it's obviously safe for a certain group of people, it's already approved by the FDA in the United States, but now they're starting to collect data on an ongoing basis from these people to really look at the impact on quality, things like quality of life and cost effectiveness. If people are on this drug for a really long time, does that reduce their disability? Are they able to stay working longer? Do they have a better quality of life? And those would be when they're, again, past, already been approved by the FDA. Now, as I said, these trials cost a lot of money. There are a lot of people that you would meet in a clinical trial. The clinical trials team, to me, starts with the participant in the center. And I really try not to say patient because you are a partner in a clinical trial. You are receiving an intervention, but you're not necessarily getting, it's not clinical care. I made that distinction already. The sponsor is very critical because they provide the funding, and that sponsor might be the setting that you're in, it might be the clinic itself, but more usually it's either a federal funding agency like the National Institute of Health or a pharma company or a device company that's creating a trial. So it could be like a Biogen or a Sanofi, some of the people that sponsored this, or it could be somebody like Hokoma, who's made a Lokomat or a walking training device. There's the research organization itself, and I mentioned it could be an academic setting, it could be a university, or it could be a clinic like where I work, Shepherd Center. We're not an academic setting, we're not a university. We are a clinical hospital providing care to people with MS, spinal cord injury, brain injury, but we have a research institute within our center. Investigators are the people who are running the trial. Then there's the pharma or device developer who is a partner in this trial. Not only are they oftentimes providing the funding, but they're also providing the intervention. Oversight entities would include things like the Federal Drug Administration, the FDA, and then there's something else I'm going to talk to you about called the IRB. And then that should say other stakeholders, not takeholders, and consumers. So that might be your care partners, it might be community providers who might be engaged in the trial in some way. So a participant is the human being, always important to remember, who's participating in the investigation, either receiving the intervention or as a control participant. They might be a person that does not have any condition, any disease, or they might be a person with a disease. Typically in an MS trial, we'll want people who have MS, but haven't had another neurologic disease, may not have heart disease. In an exercise trial, I don't want people with broken bones in the muscles that are going to be exercising. So they are otherwise healthy, but they may have MS. But importantly, you should always, as a research participant, think of yourself as a research partner and be treated as a research partner. You have a lot of responsibilities. You are signing up to participate throughout the entire study because your actions in that trial influence the outcomes. So not showing up on time for your testing or not taking the drug when you're supposed to or doing the sessions as many times as you're supposed to has an impact on the trial. And every time that happens, there's a cost to the trial. So you are a partner, you're an important partner, and you have responsibilities. If you have adverse events, a negative consequence during the trial, you are to share those. It might be that you got sick, and to you, it doesn't feel like a big deal. It's just a cold, but maybe it impacts how the drug affects you. Maybe it impacts your energy during your exercise. So you're responsible for telling your study investigators what's going on. You're supposed to adhere to the program so that we can all feel confident at the end. And that program, it could involve different things. It might just be, doing a diary, of what you've eaten throughout the day for 2 weeks. It might be doing the exercise program twice a week and checking in with a coach. It might require you to travel to the site once, 3, 4, 5 times during a trial period, which means that there might be time away from work or home. And you need to know about those things before you commit to a trial. And it may require ongoing communication. For our exercise trial, in the beginning, there's a weekly interaction with the coaches. With a drug trial, it might be talking to a nurse coordinator about what you're experiencing with the drug. The principal investigator is the person who is over the whole trial, oftentimes called the PI. They supervise and conduct the study. They tell people about what's going on in the trial. They are required to report any adverse experiences that their participants have to the sponsor or the funder. So whether that's the pharma company, device trial, or NIH, the National Institute of Health, they have to inform. So they have to maintain very distinct records, adequate records, and make them available to the sponsor. They have to ensure that they get approvals from what's called the IRB, which I will tell you more about. So they are responsible for everything. So they have to ensure the safety of the participant, and they have to ensure that all of the study protocol is followed right to the T. Every trial has a coordinator, and the coordinator, some would say, are almost as important, if not more important than the PI, because they make everything happen. They're the boots on the ground. They ensure that the PI, the principal investigator, manages every aspect of the trial and that all the activities take place. They're the ones who enroll the participants and take them step by step through the trial. They might be helping to coach the investigators and the rest of the study team. Our coordinator for our exercise trial interacts with our coaches that are providing the exercise training. They help shepherd the participants through the whole trial, and they work with the people who are doing the measures, the evaluators, to make sure that they get scheduled at the right times. So they schedule and conduct visits. There's a lot of documentation that has to happen, information about the participants, information about the measures that are collected. That all has to be documented. A lot of that now is documented electronically, but a source document is the document that contains all of that data. They have to make sure that they're supporting the participants or the subjects throughout the entire trial, and they make sure that they recruit the participants, they enroll them in the trial, which means get them registered into the trial, and that they retain them in the trial. Because every time we lose a participant, that costs money and slows down the trial. So our study coordinators, they always look this happy. They interact with everybody, departments, individuals, participants, the study sponsor, and the different committees. So they're a very important part of our trials. Now, I've mentioned a couple of times the Institutional Review Board. It is a critical component that protects, at the end of the day, their sole purpose is to protect the rights and welfare of human research participants. They make sure that the risks are minimized and that the benefits outweigh the risks, or that the risks are reasonable in relation to the specific anticipated benefits. The risks need to be worth the information that is to be gained. They want to make sure that the selection of participants is equitable. That means that anybody that meets that criteria knows about in a reasonable distance, knows about the study, and is equitably considered to be in the trial. So there should be no race and ethnic or other issues that would limit their ability to participate if they meet the criteria. Now, to be sure, there are trials that might say we're only recruiting Black Americans because we're very specifically interested in finding out what happens for them in this experience. But anybody who meets that basic criteria should be eligible to participate. And a PI cannot say, we're not taking that person because they come from South Georgia. They can't make subjective determinations. The IRB ensures safety and privacy of all data. The data confidentiality is maintained. So that means that any data forms don't have identifying information that would allow somebody else to know about somebody being in a trial or know more about that person than is really necessary for the study to be conducted. And if there is a vulnerable population, that they're protected. If we enroll somebody who has cognitive deficits, problems with thinking or memory, and they might be compromised because of that, we have to make sure that we have actions in place to protect them. Some studies you will see, especially drug trials, will say that they won't accept women that are pregnant because there's concerns that that drug might impact their unborn fetus. One of the other very important considerations is that people, when they enroll in a trial or consider to enroll in a trial, they should be fully informed. And there is a process that you go through when you are considering being in a trial called the informed consent process. It starts with the first contact and it continues until the study is completed. So an informed consent process involves, there's a written document that gets approval from the IRB, and it gives the participants all the information, the purpose, the duration, the benefits, all of the things that I just told you have to be provided by the informed consent. And that is the time at which you should be asking questions and ensure that you understand every aspect of the trial. I see Deborah came on. Deborah, do you need me? Okay.
No, I'm listening. I see when you're all done, we do have a question that popped in that I wanted to be able to run past you.
Okay, great. Should I continue and then ask it? Okay. So why enroll in trials? It sounds like a lot of work, right? But they are put forward and really carefully considered by the sponsors and the funding agencies for their relative importance. They address very important questions that are very relevant to us. And so at the end of the day, it's listed as number 3, but of course, one of the elements of enrolling in trials is you're helping to advance science. You're helping to advance the care for people with MS. But other benefits are that you, in fact, may receive a treatment before it ever becomes available to the general public, or be part of the process that helps determine if that treatment would even be right for you. You may be involved in something that provides relief or a cure. You know, people who engage in rehab studies, oftentimes they're getting something that they might not be able to get when they're out in their, you know, in their normal community, getting their normal care because insurance may not reimburse it. Many trials will provide a stipend so you can earn some extra money. And oftentimes when there are trials being done somewhere, you can consider that you're probably getting some elevated medical care. Not that the other places aren't providing good care, but by virtue of having access to those trials, having access to that way of thinking, that might elevate the care that you're receiving. What has come of all these kinds of trials? Here is a beautiful timeline starting in 1993 with the very earliest treatments, disease-modifying therapies for MS, interferon beta 1b, and showing you over the years all the different disease-modifying therapies and drugs that have gone through clinical trials and ended up being delivered to patients. The reason they can be delivered is because they went through those phases of trials. They were rigorously studied. We know that they're safe, and we know for whom those are actually going to be effective. The same is true. This is how we found out about biomarkers, is by carefully studying the brains of people with MS and looking at comparing, as you can see here in this figure, controls to people with clinically isolated syndrome, early symptoms and signs of MS, to those with relapsing remitting to secondary progressive MS. So now we can use that as a tool to see if we can change that with different drugs or different interventions. And finally, in my world, there are exercise guidelines that were developed out of rigorous clinical trials evaluating exercise interventions in people with multiple sclerosis, so that we know we can recommend they need to get 30 minutes of exercise twice a week, moderate aerobic, as well as strength training exercises. Only by getting people to participate in all of these trials do we understand more about how to detect MS, how to treat MS, and how to help reverse disability. There are lots of different resources. One of the best is clinicaltrials.gov for finding trials. I actually was going to show you how that works. If I do still have another minute. Am I good?
Yeah.
Okay. If you go to clinicaltrials.gov, oh, maybe it's not going to do it. Oh, here we go. Oh, I don't think you can see it. Can you see this? Did my screen change?
Yeah, it did.
Okay, so this is clinicaltrials.gov. You can go into it and you can put in what you want to search. Multiple sclerosis. You saw that you could even choose what type of multiple sclerosis. You can choose if you want to look for recruiting trials. You can look for different age groups, and you can look for different phases. Maybe you don't want to do phase 1 because you are concerned about safety. You're willing to consider a phase 2 or a phase 3 trial. You can apply, and then you can find out what studies are going on. You can see what kind of interventions they're looking at, looking at and where those studies are occurring. So that's a very nice resource for you, clinicaltrials.gov. You can also go to things like a clinical site like ours. I put here shepherd.org. You can go to our website and look for our research trials, but that's true of lots and lots of academic facilities and MS centers that are engaged in research. They will have a place on their websites that tells you what trials are going on there. And then also the National MS Society has the trials that they fund. So I just wanted to give you some examples of finding ways to participate in trials. So clinical research, it's conducted in humans, it's done in a rigorous fashion using a scientific method so that at the end of the day, you can make a decision about whether what's being done is safe or effective. We want to make sure that participants are always safe, that the data is good integrity, and that you as a research participant know that you're a research partner. So with that, I'll take any further questions. Thank you.
Incredible information. Thank you. We did have something from Gillian, and you sort of touched on that because I think he can use the system you just showed us as to where to reach out to, but Gillian lives in Trinidad. And I know we have some participants that live in Canada. Some of them live in, I think we had Australia one time. So can they go to the same places and ask for information or get involved, or where do they have to go?
They can go to the same places. I know for us, we are not doing any trials in other countries right now, although we're part of clinical trials that are, and that would be on clinicaltrials.gov. I think I know that you can also find out through, I was going to say through Can Do MS. They also have some people in Canada, and I think they also share information about things that are happening in other countries. But clinical trials is a good place, and the MS Society funds international trials as well. It's a good question, Gillian.
Yeah, I know that we also have somebody that hasn't said anything yet today, but David comes from, I think, the Bahamas, and he generally comes to all of these programs. And I'm sure he would, if considering that, would need to know where to go also. So
yeah,
good. Thank you.
Mm-hmm.
Okay, so we have a couple more minutes. I don't know if anybody else has a question, but we'll hold off for a few seconds and see if anything comes in. Casey, I don't know if you have any questions offhand that you want to ask or not. I'm going to give you that opportunity the same. I know you always come up with things that I would never have thought of. So in case you have something, now's your chance too.
Way to put me on the spot, Deb.
I know that you have things in the back of your head. I mean, she answered everything already. You really were so
That was a very, very thorough presentation.
Very, very thorough. So I'm sure nobody has any questions to ask because you've answered it already.
Well, I welcome people to reach out. I'm fine with people emailing me if they have questions. I just realized that's an old email. It's Deborah.Backus@shepherd.org. I have no idea why it's the old one, but
honestly, if anybody that's listening has a question that they think of, you can respond directly to the email that you got reminding you of this presentation, and we'll make sure to ask Dr. Backus that question and get back to you. So that can work also. Okay, so well, that brings us to the end of our time. If you missed any part of this conference, it was been recorded and will be available through the MS Focus Facebook and YouTube channels. Reply to your registration email for information on how to access recordings or sign up for our newsletter to learn about upcoming events. Our next National MS Education and Awareness Month teleconference will be Thursday, March 18th. That's in 2 days at 5:00 p.m. Eastern. MS Certified Nurse Practitioner Katrina Bodden will be discussing everything you need to know about MS. And our sincere thanks to Biogen, Genentech, Novartis, EMD Serono, Sanofi, Viatris, and Bristol Myers Squibb for their generous support. and all of our attendees for your participation. A special thank you to our guest speaker, Dr. Deborah Backus, for taking the time out of her busy schedule to share this important information with us. We greatly appreciate each of you filling out our post-event survey, which will be available as soon as you leave this meeting. Your participation in the survey will help us greatly in creating meaningful programming for the MS community. Goodbye, everyone, and we look forward to seeing you again soon. Thank you, everybody.